All right, so Oksana and Ben, we're
Okay,
gonna
Abe says
start
he's ready
now.
to get started.
Yeah, we're time to get started. You gotta quiet down the conference room for me.
All right, everyone. I think we're
All
gonna
right.
get started.
I'm happy for everybody who's joining us in person and online. Just for those of you that didn't get the memo, it turned out that on Wednesday I tested positive for COVID. And so although I was really looking forward to this as an in-person event, I'm sitting in my office 2 floors down from the people who are there in person and won't be able to join you in the conference room. But I will be sitting out on the porch on level 2A during the breaks. And anybody who feels brave and wants to be around somebody who has COVID when they're sitting outside is welcome to visit me there. I'll be at lunch too. There's an outside area for lunch. Okay, so this is the 10th time that we've done this, the 10th time that we've had a retreat for this center. And I really appreciate the people who've come to join us. Some of you are brand new and some of you have been with us for all 10 years. I did want to give a little bit of a historical perspective on where we've been and where we're going. So originally we submitted this grant in 2013 in September, so almost 11 years ago, and it was funded. And then about a year or two after it was funded, I moved to Chicago. So in a sense, one important component of the project moved from Chicago to UCSD, to San Diego, where we are now. And then Leah, who, as you'll hear in the subsequent slides, if you don't know already, moved in 2018. She was in Milwaukee and she moved to Wake Forest University in North Carolina. We were able to renew the grant in 2019 after the first 5 years. And that renewal is now in a no-cost extension. Technically, it would have ended a few months ago, but we have a 1-year no-cost extension and we have a little bit of money left with that. But obviously, to keep going, we had to figure out how to move forward. So it turns out that NIDA P50 center grants can only be renewed once, which, as I just explained, we did in 2019. And so we couldn't just submit another renewal of the P50. Different institutes have different ways of handling P50s. And so instead of a renewal of the P50 last fall, about a year ago, we submitted a P30 grant. And as I'll explain in the subsequent slides, a P50 and a P30 are similar. They're both center grants. But a P50 has both cores and projects, whereas a P30 has only cores that are intended to support existing grants. So it's a little bit of a weirder animal. Fortunately, we got good reviews for the P30, and then just last week, we finally got the Notice of Grant Award, which if you do this for a living, you know they always make you wait months and months on pins and needles to find out if you actually get the money. But we actually got the money. And so the good news is that now we're funded through May 31st of 2029, which is a shockingly far in the future, especially if you were born on the year I was born. It's way in the future. So that's terrific news. Very happy to have that support. Again, just from a historical perspective, originally we conceived of this as a center that had a breeding core that produced rats, heterogeneous stock rats. I'll talk about them in subsequent slides, and sent them to 3 different projects that were essentially phenotyping projects. And then those projects would send the DNA to a sequencing and genotyping core that was originally in Chicago, but as I explained, moved to San Diego. And then we had a whole project that was about developing the methods to, or refining really the methods to analyze the relationship between genotype and phenotype to perform genome-wide association studies, which I'll be calling GWASs. And those were supported by an admin core and a pilot project core. In the renewal of that, we shuffled it around a little bit. There were slightly different phenotyping projects, but there was still the core of the structure was that we produced rats, we phenotyped rats, we genotyped rats, and we analyzed them. We had gotten good at doing the kind of basic GWAS analysis, so we added a research project that was the PI was Trey Eidecker here at UC San Diego, that was using a network-based approach to try to extend on the findings from the GWAS and also to integrate them with other species, particularly human. And that was productive, but as you heard, is sort of sunsetting now. Very significantly, right about the time that we originally got this P50 grant back in 2014, NIDA came out with this U01 mechanism, and they've reissued this 4 times, most recently reissued it just in the last few weeks. And this has been, they've had different names, but essentially it's the Animal Genetics and Genomics U01 mechanism. And it's a special call for grants that use animal models to study the genetics relevant to drug abuse. And it's been incredibly helpful, I think, for this center, because as you'll see in the next slide here, what we really had, and this was as of a year or two ago, what we really had was a core that produced animals. We had some projects funded through the P50 that phenotyped them, but then we had numerous additional projects that were funded by this UO1 mechanism that were also phenotyping animals. And many of the people in the room are familiar, if not responsible, for these different UO1 projects, as well as at least one R01, I think now two R01s that are using HS rats for phenotyping. But so that UO1 mechanism has really been essential. And I think if it weren't for the UO1 mechanism, we wouldn't have been able to get the P30 grant, because as I said a few slides ago, a P30 is a weird sort of a grant. You only have cores, and you've got to explain what already funded projects are being supported by those cores. And so what we were supporting were many different UO1 grants, as well as R01s, K99s, et cetera, even a UM1, which is one of these Scorch proposals, the HIV, single cell, et cetera, proposals. But really the core of the grants, or one of the cores of the grants that we said we could support as a P30 center, was these different UO1 mechanisms. And so again, the P30 is a similar structure. It produces animals. Leah's going to talk about that. It genotypes and analyzes animals. I'll say a little about that in Pejman. We'll say more about that. It has an admin core, which I'm going to talk about in just a few slides, and a project core with Oksana we'll talk about in about 40 minutes. But it's now entirely in support of separately funded projects, which is a weird sort of a grant to write, because you're supposed to be innovative, but at the same time, what you're doing is activities that are core activities as opposed to research projects. Anyway, we managed to thread the needle on that, and as I said, I really think it's because of the UO1s was a big part of that. As I was writing, though, this grant now a year ago, the P30 grant, I had originally thought of this center back in 2013 as a center that was going to do GWAS. It was going to do genome-wide association studies. So we were going to phenotype animals, genotype animals, and look at the relationship between genotype and phenotype. But as I thought about what we wanted to propose as the P30, I realized that that was probably too narrow, and that the center had already become about more than just GWAS. And that really it was about providing the field, the drug abuse in particular field, with a platform for studying individual differences. And that meant that we needed to be able to produce diverse animals, genetically diverse animals. It meant that we had to be able to genotype and analyze those diverse animals. But having those populations has opened up a lot of possibilities beyond GWAS. And one of the obvious ones is people looking at gene expression, epigenetic studies at the single cell level or at the bulk level. But there are other kinds of projects and grants that have grown out of that. And so when we put in the P30, we actually renamed it, and it has a name. I don't think I have the name right in front of me, but it's sort of Genetics, Genomics, Epigenetics. It's a much broader name, whereas previously we had had the word GWAS actually in the name of the center. And that really reflects what I think is the evolution of this project. And it's really gratifying to see that over time, it's grown to be sort of wider in scope than what it originally was. I was just at a Gordon conference, actually in Alyssa's home state of Maine, and you can't take any pictures at Gordon conferences, so I'm not able to show you a photograph, but over and over and over again, the people at this Gordon conference, which was about drug abuse, but not specifically genetics, would put up slides like this one here, which I grabbed off the web, where they would talk about brain regions that are important for addiction and the relationship of those brain regions to behaviors that are important for addiction. And I do think that one of the really defining things about this center over the years is that we've been able to support projects that are studying these different phases of the drug abuse cycle, and there are different versions of how this circle looks, but here's one that's illustrative. And they have things like delay discounting, which we're explicitly studying in this project. They have things like Q reactivity, which we're explicitly studying. And so I think it really is an example of how the resources that we've been able to provide have allowed people to study in sophisticated and quantitative ways these essential aspects of substance use disorders in animal models, which of course have strengths and weaknesses relative to studying them in humans. So the center from the very beginning was built around the HS rat, the heterogeneous stock rat. Many people on the call and in the room know exactly what I mean by an HS rat, but I know there are some people that are newer that might not. HS rats were created in 1984 by mixing 8 inbred rat strains, and they've been maintained as an outbred population now for more than 100 generations. And by outbred, I mean they're mixing every time. They're not brother-sister matings. And so we're breaking up these big haplotypes that existed in the original inbreds here that are shown as solid colors into littler and littler pieces. And that's important because as they get broken up into little pieces, a genotype that's characteristic of one of those different colors will only be associated with a phenotype if it's extremely close to the causal allele, the thing that really is influencing that phenotype. And that allows us to get precise mapping of the relationship between chromosomal regions or genetic differences and phenotypes. That's the essence of what GWAS is. I have Leah's picture here to remind me to say that Leah is entirely responsible for the availability of the HS rats to this project. So when we started putting together this center 11 years ago, Leah was already maintaining in her lab a colony of heterogeneous stock rats. She's continued to maintain that in support of this project the entire time and still maintains it today. And she's going to be talking immediately after me about core B, which is the HS rat core. I'm just going to have 1 or 2 slides about it. Right now here in San Diego, and I think also in North Carolina where Leah is, the 103rd generation of the HS population is being born. And it's important to say that this center is the only, maintains the only colony of heterogeneous stock rats in the world. So the availability of these animals is entirely dependent on this center. And possibly that was one of the compelling things that we were able to talk about in the P30 grant. So an essential feature of the center is maintaining and distributing these animals. Here's a slide. This is a poll from a tool that I'll talk about in a few minutes called IRS. And this is a poll of all of the animals that have been cataloged in our database, all the HS rats that have been cataloged. It's too small to really see, I understand, but the take home is there are about 22,000 animals now that have been produced and used by this center. So it's really a huge population. And one of the things that I've been realizing over the last several years, and this is based partially on feedback we got from the external advisory board, is that the heterogeneous stock rats, originally we were excited about them for GWAS as a tool for GWAS, but really they're an outstanding tool for anybody that wants to study individual differences, anybody who wants to study the relationship between genotype and phenotype. And there are a lot of labs that are interested in individual differences, but right now they're working with commercially available populations like Sprague-Dawley and Wistar and et cetera, which are not maintained very carefully. We've actually published a paper, particularly on Sprague-Dawleys, where we looked at what different vendors call Sprague-Dawley, and they're incredibly different from one another. And in fact, the different breeding facilities are extremely different from one another, and even rooms within the same breeding facilities are different from one another. And when people purchase animals from these commercial vendors, they're not often aware of these differences, and it can badly confound the results of studies. And we talk about that in that paper, and I'd be happy to follow up with people. But it's really a very serious problem that's leading not only to irreproducible results, but also can drive false positive results. And so one of the things that I really hope we'll accomplish in the next 5 years is promoting people that don't necessarily want to do GWAS studies to nevertheless consider using heterogeneous stock rats when they're interested in looking at individual differences. Leah's going to cover this also, so I don't want to dwell on it, but we did about a year ago set up a colony of heterogeneous stock rats here in San Diego, which we're calling HS West. It's a satellite colony of Leah's colony. We still think of Leah's colony as being the main colony. Most recently, Yiji, Rudy, and Nina in my lab are maintaining that colony. And we did that because we, although initially we didn't have any trouble shipping animals, Leah didn't have any trouble shipping animals, starting a little before the pandemic, we did start to run into problems where the commercial carriers were screwing up and in some cases killing shipments of heterogeneous stock rats. So Leah increasingly has been trying to use ground shipping instead of air shipping. But with a number of projects funded here at UCSD, it's really impractical to ship by ground from North Carolina to California. And so we set up a colony here, which has been feeding those local UCSD projects. And we are actually taking steps that I think Leah will talk about to maintain, to pass animals back and forth between the colonies. I also want to briefly put in a plug that working particularly with Hao and Mindy, who generate a lot of data about inbred strains, other rat inbred strains, we've identified really good surrogates for 6 of the 8 founders of the heterogeneous stock. These are modern, available inbred strains that are extremely genetically similar to the original founders. And that means that people that want to know whether or not there are genetic differences for a particular trait can order these strains and use them to determine whether or not there are genetic differences among the founders' chromosomes as a way of determining whether or not they're likely to see genetic differences for their trait when they use heterogeneous stock animals. And that's really important and useful. And there's a project that Oksana is doing with some other people here on campus that are interested in hearing, where they are actually ordering these animals and looking at the hearing of these animals as a way to get preliminary data to establish the heritability of these hearing traits. And that's something that I think some other labs are doing, but I really think it's a great first step for people that are interested in getting involved in heterogeneous stock animals, but need preliminary data to kind of establish heritability. And then also, and I believe Den is going to be talking this afternoon about this, and so I'll only mention a slide, but we do have a UO1 grant that we got funded several years ago, the goal of which is to look at other kinds of genetic variants, so not just SNPs, but also tandem repeats and structural variants within the heterogeneous stock. And that's led us to generate short read coverage, Illumina coverage of greater than 30x for the founders. We got DNA from 1984, so what we sometimes refer to as ancient DNA. And we've also generated a lot of PacBio coverage, about 40x for the founders. And most recently, we've generated Oxford nanopore ultra-long reads, so greater than 100 kb reads at 40x coverage. We were only able to get that from 5 of the 8 founders because of DNA quality issues. And we're now working with a core here at UCSD to get high C coverage on at least the remaining 3. And Den will talk about our use of that to discover different kinds of features besides just SNPs. But it also means that in the future, we think we'll be able to use that data to put together a pan-genome and potentially reanalyze all of the accumulated data over the last 10 or more years in a pan-genome context, which may have some advantages. Another essential function of the center is to provide a centralized database, so to store all of the phenotypic, genetic, and other kinds of data that we have about these animals. Originally, when we wrote the original center, I imagined that as a half-time position, and Apurva Chitre in the lab was the person initially in charge of the database. It's ballooned. I really underestimated how much work it was to do that well. It's ballooned, and we have a team currently of 4 people shown here: Tiago, Ben, Elaine, and Gabby, who run the bioinformatics thing, which now we've merged into Core A. So we now say this is part of the admin core, but these are the people that maintain all of the data. And the database uses FAIR practices. We have portals making the data available both on rat genes and rat GTEx. And we have assisted a number of people with data management and sharing plans that the NIH now requires, although to my knowledge doesn't really do much with, but one day they may do something with. And so we're happy to help people who are writing grants to identify the best ways to share data. We do a lot of quality control. So a big function of this is really to take in data from different projects and to filter the data, do QC on the data. I think Montana may talk about that a little bit in her talk this afternoon. We have worked a good relationship with the UCSD library, which hosts the data and will maintain access to the data in perpetuity, they say. And so we put data there, larger data, and particularly sequencing data goes on SRA. We have GitHubs to describe some of the pipelines. DOIs are generated about computational pipelines. We've got registered RRIDs for all of the animals that we have. And then most recently, we have the IRS, the Internet Rat Server. And this has really been a brainchild of Elaine, who did this and has come up with a logo for it. So the IRS is a new way that we are interested to get feedback about that different people involved in the center can query our database and get back information that they need. So here's kind of a schematic of all the kinds of data that we have. In the interest of time, I won't talk through it. But she's developed a few tools here. One of them that comes up a lot is locus zoom plots. So when somebody has a GWAS hit, we give them a plot, often with about a 3 megabase window, showing them the exact LD relationship and p-values of SNPs within that window. And I think you'll see a lot of those plots as the day goes on. But sometimes people want to zoom in or zoom out, and it's been a kind of a exchange of emails that's been necessary to do that. This allows people to just go to this website and to produce locus zoom plots with different levels of zoom and other properties, and that's something we want to get feedback on. We also have created a tool, or rather Elaine has created a tool to do PheWAS studies. You can look at the gene level or based on coordinates, and you can identify other traits that have been associated. Basically querying a table that we have of GWAS results. And you can also pull rat metadata. So if you want to find out, who begot who, like who are the breeding relationships, the sex, the date of birth, et cetera. And soon we'll have all phenotypic data queryable here. This section of it is going to be under a password so that only people who should have access to different data sets can access those data sets. But this is something we're rolling out today at the meeting, and we would love to get feedback from people about whether this meets a need. And it's certainly useful for us internally, but we think it might also be useful to a bunch of our collaborators. The data from the center is actually attracting a lot of interest from the growing group of people that want to do artificial intelligence and machine learning and are looking for available data sets. And of course, one of the advantages of rat data relative to human data is that it's really easy to share. I'm not going to say more about that, but there have been a number of inquiries, and I feel like it's accelerating, and I think that's great. That's another kind of ancillary benefit to the center. Another essential function of the center is to provide an expensive way of genotyping animals. And so that's been a function of Core C since the inception. Here I've got people that are currently involved in that: Kai, Ben, Den, and Gavi. I don't think I'm going to say a lot about this except to say that we've changed a little bit the sequencing component of that. And that created a little bit of a complicated situation where we had to produce genotypes that were consistent regardless of the way the animals were sequenced. And that was something that Dan and more recently Ben and Gavi have worked on. And we have now a hybrid genotyping pipeline. Doesn't matter to most of the people in the room, but we have a very reliable way of producing genotypes. And we actually produce genotypes that are about 99.75 or greater accurate compared to just 30x sequencing of the same animals. And there's a paper now that's under review. I think it got good reviews describing that pipeline. And then the final essential part of the center is the ability to relate genotype and phenotype to perform GWASs and other kinds of analyses, some of which Pej will talk about in his talk later this morning. This is something that Apurva really led initially and that more recently Tiago has taken over. As many of you know, we produce reports, and these reports are increasingly interactive reports that allow people to look at Manhattan plots, logosum plots, EQTLs, PheWAS, et cetera, the different kinds of standard analyses that people would want to do. And these make it much easier, we hope and we believe, for people to write papers. Recently, we've added a lot of text explaining to kind of newbies how to interpret the different sections of these. And now on the IRS server, another benefit is that now you can go and visit the most current but also older versions of reports that were issued to you, again, in a password protected manner. And those are going to be available on the IRS so that they don't just exist as like emails that you got. And we are rolling out, and Bobby is working on this, doing haplotype mapping, which is more commonly done in DO mice. And we've been doing it for a while behind the scenes, but we're now going to include in the reports haplotype mapping as well as SNP-based mapping, which the latter of which is more equivalent to what people do in humans. So all of these tools, are critical, but essentially the ability to produce animals, the ability to genotype and analyze animals is the core services that are produced by this P30 Center and that have supported a lot of terrific projects. I wanted to briefly touch on some projects. So Apurva has overseen collecting locomotor activity data from, I think, maybe 6 or 8 different projects, each of which contributed, hundreds or a few thousand animals. We now are to almost 8,000 animals in this most recent analysis, which is producing really robust results, lots and lots of hits. Locomotor is an interesting case. Some of you know that I've also been involved with a, excuse me, with a project looking at externalizing, excuse me, the COVID is catching up with me, looking at externalizing in humans. And that has produced its own large GWAS. Theoretically, many people have argued that locomotor activity in animals is related to externalizing behavior in humans. It's been hard to test that empirically, but what now Brittany is working on is using the big locomotor analysis that Apurva has produced to see whether or not either the same genes are being identified in humans for externalizing and rats for locomotor activity, and if not the same genes, if the same parts of networks of genes defined with lots of different data types are being implicated by those two. And that follows up on some earlier analyses Brittany has done including looking at body weight data in animals and humans, where she did show a lot of overlap at the network level. Lots and lots of physiological labs have glommed onto this center, and in some cases, we do experiments for them or they do their own experiments, but in many cases, we're able to either measure things for them or provide them with tissue, and that's been another major ancillary benefit of the center, and that continues. It's allowed us to get a lot of, for example, body weight data that Apoorv has also gathered from many different labs. I think this is, I'm not even sure this is the most current, but here she has 8,000 animals for body weight, producing 37 independent genome-wide associations. So this is really looking more and more like the kinds of results that are coming from human GWAS, but that typically require hundreds of thousands of subjects to get to the point where you're getting dozens of genome-wide significant hits. So we're really excited to have that progress and to see the power. And then the last thing that I'm going to mention is Gattaca. Gattaca is a dystopian science fiction movie in which people learn to select embryos, and they select embryos based on genotypes that they'll have high predicted fitness traits, and then there's sort of like a underclass that wasn't genetically selected, and then there's an elite class of people that were genetically selected, and there's discrimination against the underclass that's both legal and extra legal. It's terrible, dystopian kind of a situation, and I'm totally opposed to it. But Radica, named by Paul Meyer, who's in the conference room, Radica is a wonderful thing where we do exactly that. So we genotype animals when they're young. We're able then to predict traits in the animals, and we think this is really exciting. This is something that we're offering as a service in Core B. We think this is really exciting because we can now give people animals that are predicted to be very divergent for traits, even though the animals haven't yet been exposed to any of the phenotyping procedures required to measure those traits, some of which might involve, for example, the administration of drugs that would be themselves expected to change the biology of the animals. So we can do this using very standard techniques. To do it, we need to have a large database of genotyped and phenotyped animals, which, as I've described, we have. And that allows us to predict any of the traits that have been studied by the center, but critically, I should say, not traits that have never been studied by the center. We can only do this for things we've studied previously. We have to collect the tissue at a young age and genotype the animals rapidly, which we've gotten good at doing also. And then we need to have a large population of animals to select from, so that if we select, say, the extreme high 5% high and the 5% low, if those came from a small population, they won't be that divergent. But as the population becomes large, and in our case, 500 is a ballpark number for what we look at, then we can expect to get animals that are really extremely divergent, as shown in some of these figures. We can do this better for traits that are more highly heritable, and that's what's shown in the figure on the right, is that heritability allows us to do a better job. But even for traits with pretty modest heritability, we're able to get groups that are significantly different from one another. And the other thing that influences the quality of our predictions is sample size. So as sample size goes up, our prediction gets better and better. But we find that even with pretty modest samples of about 500, we can already start to get meaningful prediction for Radica. So we now have sort of a menu. We're working on this and working with some of the people in the room to figure out what are the best traits to offer to predict on. But really, in principle, we can predict on any of the traits that have been studied by the center in the past, provided that they have decent sample size and heritability. And we think that opens up a lot of cool possibilities for new kinds of experiments. And you'll hear from both Marcita and Matt this afternoon about some things that they've done with Radica already. The very last slide, and it looks like I'm right on time, it's 9:30 now, is that I am, as I think about the health of this community, thinking about the importance of continually making efforts to diversify the topics we work on and the funding that we receive. So originally, HS Rats were funded by NIDDK, a grant that Leah had, an R01 grant that Leah had. So that funding has been ongoing for a long time. Relatively recently, both Giordano and I have gotten NIAAA grants to work on these animals, and I think Marcita is likely to get an R21 to work on these animals from NIAAA, so that's great. We've had some previous funding from some of these exotic, to me, institutes like National Eye Institute and Musculoskeleton. I think Doug is on the call. That was his grant. We got a supplement from NIA, and NIA is somebody I'm really very interested in continuing to develop our relationship. It's a bit of a sore spot to work with NIMH, but I'm eternally hopeful that maybe we'll be able to find a way to propose research that appeals to NIH's priority. We also are getting some California money, so both CIRM grant and TRDRP are avenues that we're exploring or have explored to get money from the state of California. And there was an attempt with NIHS, and we even had conversations with NASA. So a priority that I'm interested in talking to everybody about is to make sure that we're not only doing this for NIDA. The animals are terrific, and the system is terrific for the kinds of research that NIDA is interested in, but there's no reason it should be limited to that. And I think we will be, as a center, healthier and more productive if we are diversified in our topics and funding. So I have tried to acknowledge people as I went through these slides, but it literally does take a village to do all of this stuff. Here's a picture from last year. I hope we'll be able to take a nice picture this year. And here's the Radica logo. I think there's a bunch of swag for the people that are here in person as we try to promote the idea of Radica. And I should have said, but we'll say now that we plan to have a booth at SFN next month in Chicago, promoting and supporting people's interest in Radica. Okay, so that's the end of what I was prepared to say. We could take a few questions now, but I'm going to unshare my slides so that Leah can queue up her slides. Leah is also presenting remotely, and then we'll go to, I think, almost entirely in-person presentations after that. I hope you guys were able to hear me for the last 30 minutes.